Hi everyone, welcome back to Useful Genetics. This is Lecture 2C, where we're going to talk about why most mutations are harmless. And there's really 2 points. Most new mutations are not in genes, and even when they're in genes, most of the mutations in genes are in introns. Now, here's a diagram based on analysis of the human genome. We could draw a similar genome for most other plants and animals because humans are really fairly typical. So the first point is most DNA is not part of genes. Half of our DNA is the kinds of sequences that are called genetic parasite sequences. They're repeated sequences we have many copies of, different categories called lines and signs and LTR transposons. We'll talk more about these later in the course. The main point now is that they don't do anything to serve the functioning of the organism. So another 20% of our DNA here is sequences with no known function that's not genetic parasites. There's also a chunk here, about another 10%, that's repeated sequences like the genetic parasites, but these sequences don't actually look to be parasitic sequences. They're just repeats that don't have any known function. So only about 28% of our genome is genes. I've superimposed a dartboard on the drawing of the genome to make the point that we can think of our genome as kind of a target for mutations. The mutations, however, are blind. It's as if the person throwing the darts was blindly throwing darts at the dartboard, and we expect they're going to land randomly all over the genome. So naturally, most of the mutations are going to land in most of the genome, which is the non-functional parts of the genome. This segment here is going to get most of the random mutations. Now, even when we consider the part of the genome that consists of genes, most of this DNA does not code for proteins. Only about 2% of our genome actually codes for proteins. The rest of this 28% is intron sequences. You remember that introns are segments of sequence that are cut out of the RNA molecule after it's been transcribed from the DNA, and only the remaining coding sequences become part of the messenger RNA that's actually translated into proteins. So introns are also largely non-functional DNA, and that's where the bulk of the mutations that land in genes happen. They just, by random chance, most of them are in introns. It won't take long now to describe what we've done. We've considered the reason that most mutations don't cause us any harm or have any effect at all is because most mutations are not in genes. They're in the very large fraction of our genome that's non-functional sequences. Even within our genes, most of the mutations that happen are in introns. Most of the sequences in introns are non-functional, and mutations there don't make any difference. It's only a small fraction of our genome that's coding, and even within that, there are differences in how much different sequences matter. Coming up next, Lecture 2D, we'll talk about mutations in coding sequences that don't affect function. We're going to reserve the discussion of functional consequences largely to Module 3. So here we'll be talking about neutral mutations and silent mutations. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 2D. We're going to talk about 2 ways that mutations that are in coding sequences may nevertheless not change the function of the protein they specify. The first is that the mutation may be silent, that is, it doesn't change the amino acid that it specifies. The second is that the mutation may be neutral. It changes the amino acid, but that change does not interfere with the protein's normal function. Now, in the previous lecture, we talked about why most mutations are neutral, because they're in the very large fraction of the genome that either is not part of a gene or that is in the introns of genes. Now we're thinking about the 2% of the genome that does code for proteins. Even in here, many changes don't change an amino acid. Now, because this depends on the properties of the genetic code, I just wanted to take a minute to talk about how we display the genetic code, because it's not obvious if it's not explained. So you've seen, I know you've seen because I showed it in Module 1, displays of the genetic code like this, where there's this complicated table with letters on all sides. It's hard to figure out what's going on. Really, tables like this are just a more compact way of displaying a list. So you can consider each part of the table as being part of a list. So we have listing the entire genetic code in this list. That's easy to understand, but it's pretty inconvenient to represent in a picture on a page. And so it's usually assembled into this more clever table diagram. The basic structure of the information, though, is that the 3 bases on the left side are 3 bases that form a triplet in the messenger RNA. And they will be translated as instructions to put a particular amino acid at this position in the protein sequence. And the 3 letters on the right are the abbreviation of the name of the corresponding amino acid. In this case, GGG specifies glycine. Now, there are other ways to represent genetic codes. Molecular biologists kind of like making things clever. So, for instance, you can represent it as this complex circular diagram instead of a table. This is more elegant, but I don't find it easier to understand. Now, back to the question, why most mutations in coding sequences are neutral. And the first part is that many changes don't change an amino acid because the genetic code has the property of being highly redundant. Redundant means you've got more than you need. In this case, it means we've got more information than we need. The genetic code could, in principle, specify 64 different things because there are 64 different ways to put 3 amino acids, 3 bases together in different orders when you've got 4 different bases to choose from. But there's only 20 different amino acids. So the result of the evolution of the genetic code has been that most codons, most amino acids, such as valine, are specified by groups of codons. Here, 4 related codons, they differ in their 3rd position, all specify valine. Many other amino acids also are specified by groups of 4 codons. Some are specified by 3 or 2 codons, or leucine, specified by 4 codons. Only a couple of amino acids have a unique codon specifying them. Now, what this means is that it's possible to change the codon, to change the triplet code in the net messenger RNA without changing the amino acid that's inserted. And that's shown diagrammatically here. So here's a schematic drawing of the effect of different mutations on which amino acid is inserted. The green line here represents a messenger RNA, and we've highlighted a particular codon, particular triplet, the base is GAC, which the genetic code table tells us instruct the ribosome to insert aspartate, the amino acid aspartate, at this position in the growing chain. On the left, we've got a transfer RNA. In particular, we've got the transfer RNA that carries aspartate. On one end, it has 3 bases, the anticodon, that can form base pairs with the codon in the messenger RNA. And on the other end, it's loaded with the amino acid aspartate, ready to insert that into the growing protein chain. Now let's consider the consequences of mutations at this position. First, let's consider a mutation that does change an amino acid. So consider a mutation at this position, A here, that replaces the A with a C. Now this mutation wouldn't have happened in the messenger RNA, would have happened in the DNA, but the consequence is that a messenger RNA is made that contains a C instead of an A. What's going to happen? Well, we look at the genetic code table and we see that the mutation has changed that codon into a codon that specifies alanine. This mutant messenger RNA, therefore, will not accept this transfer RNA at all because it can't form the correct base pairs in the anticodon. And so instead, it will form a codon-anticodon pairing with a different tRNA, one coding for alanine. Now, what about a mutation that is in a different place that's silent? So here we're considering a mutation at the G. It's replaced the C with a U. Of course, in the DNA, that would be a T, but in the messenger RNA, that's a U. So now the new codon is GAU. Look up here, we've gone from GAC to GAU, but we haven't changed the amino acid that's specified. It's still aspartate. And in fact, this messenger RNA will still pair with the same transfer RNA because many transfer RNAs that code for amino acids with redundancy are actually the physical manifestation of this redundancy is that the third position anticodon base pairs quite flexibly with the third position codon base. And in fact, the G of this transfer RNA will pair with a U as well as with a C. So that's one way that changes to coding sequences don't change function because many of them don't change the amino acid at all. The other way is that amino acids may be changed by a mutation, but this may not change the protein's function. This is seen when populations are examined. It's thought that at least half of the non-silent differences in DNA sequence between different people are actually functionally neutral. They don't change protein function. There's an example of this here. This is data from an analysis of a protein called SRC. This is a regulatory protein that regulates cell growth. And what these authors did was they created mutations at many different places in this protein, and then they tested them to see if they changed the function of the protein. And they put red and green dots onto the structure of the protein to show us where changes made a difference and where they didn't. All positions with red dots are positions where changing the amino acid changed the function of the protein, usually making it work less well. The green dots are positions where changes to the amino acids didn't change the function of the protein at all. Now these are only the places that they tested. There would be many more places that did change function and many more places that didn't change function. So what we've done, we've started with what we already knew, and we've considered silent mutations that change the DNA sequence of a coding region but don't change the amino acid that's inserted, and neutral mutations where a different amino acid is inserted but it doesn't change the protein's function. Now, there's still the section of the protein where changes make important differences that affect phenotype. We'll talk about these quite a bit in Module 3. But coming up next, we're going to think about a particular category of non-neutral changes in coding sequences, and that's the changes that are called frameshifts. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 2E, where we're talking about a particular kind of mutations that cause serious defects in coding sequences. These mutations occur when DNA polymerase mistakenly inserts or deletes a base instead of just putting in the wrong base. And the consequence is that the reading frame used for translation into amino acids is changed, and the resulting protein is basically garbage. Now, we talked about replication errors by DNA polymerase that have usually little or no effect on function because the wrong base either doesn't change an amino acid or it changes an amino acid that's not important for the protein's function. So that a substantial fraction of these mutations, even in coding sequences, are functionally neutral. But DNA polymerase can make another kind of mistake, a mistake where it inserts or forgets to insert a base. And this causes a frameshift mutation. And because the new reading frame hasn't been subject to natural selection, it's effectively, functionally, it's garbage. And so these kinds of mutations are not neutral. They're almost always a complete loss of the function of the protein. Now, this is a repeat of the diagram that I showed in Module 1 to give you sort of a text-based intuition about DNA and RNA and protein. So we'll think about how frameshift mutations work in this context. So the top line represents DNA coding for a gene, and it's got two start code signals, one for transcription, one for translation, and two stop signals, one for transcription and one for translation. And in between is the text that codes for the function of the protein. The transcription line, this is a messenger RNA. This is text representing a messenger RNA. It was begun using this external start signal. So the transcript includes the internal start signal and the internal stop signal. These will be signals that control translation. Below this, we've got the same sequence broken down into its triplet codons, which here words are serving the analogous function to triplet codons. And below, we have the protein that this messenger RNA will be translated into. In this case, we translated the fat cat ate the big bad rat into Korean. Now, what would happen if there's a frame shift in the DNA? Well, here's our frame shift. We've added a C here, and the only effect is that the DNA is one base longer. Not a big deal. It doesn't affect transcription. The start signals and the stop signals are unchanged. It's transcribed normally. It's just one base longer. The problems arise in translation. When we parse the text into its three-letter words, we find that suddenly we go from making sense to gibberish because we're reading the words in the wrong reading frame. And this, of course, means that the protein that we produce from this sequence is gibberish. This is this sequence translated into internet symbols. Only the sequences before the frame shift are normal. This protein, of course, is not functional, and so the rat runs away. It doesn't get eaten by the cat. It's also frequently shorter than the protein that should have been made because very often these unselected reading frame triplets include triplets that specify stop codons. Now, let's think about what would happen if instead of a single base being inserted or deleted, DNA polymerase made a slightly bigger error. What if instead of inserting a single base where it shouldn't have, it inserts two bases? Well, the reading frame's still going to be shifted. It's going to be a different frame shift. It'll be reading the third possible reading frame, but it'll still be out of frame. What about, though, if it inserts three bases? Well, actually, three bases is just fine. the reading frame is going to stay the same. There will be a 1 codon insertion or deletion, but the rest of the protein will be the same. And it may be that inserting 1 amino acid or deleting 1 amino acid from the sequence doesn't have a dramatic effect on its function, whereas a frameshift always has a dramatic effect on function, almost always completely eliminating the function of the protein. We can apply the same logic to 4 bases or 5 bases. They are, like 2 bases, going to produce completely non-functional proteins. So what we've done is pretty simple. We've thought about one thing, frameshift mutations, how mutations that insert or delete a base will shift the reading frame of coding sequences. This causes the wrong amino acids to be inserted not just at the site of the frameshift, but at all the positions downstream from there, all of the positions that the ribosome moves through past the frameshift mutation. Very often, the frameshift means that the reading frame that's being used, the wrong reading frame, is going to contain stop codons in unexpected places. So usually these defective proteins are not very long because pretty soon the ribosome hits a stop codon. And this effect applies not just to single bases inserted or deleted, but to any insertion or deletion that isn't a multiple of 3. Now, coming up next, we're going to think about another class of mutations that can have serious functional consequences. And those are mutations that are not in coding sequences at all, but are in other functional sequences in the DNA. I hope to see you there.